All right, hello folks. We're going to talk a little bit about fertilization. This one's going to be a bit short and sweet because it's a fairly straightforward topic, and we're going to focus in on the things that are really high yield, which is really most of it. Okay, so in simple terms, you don't really need to know anything about oocyte preparation for fertilization. It sort of happens sort of incidentally over the course of meiosis in the oocyte. What we do care about is spermatocyte preparation. So basically, in simple terms, when a sperm enters the epididymis, it's fully formed, but it's not ready to actually fertilize an egg. If you take it directly from there and you introduce it to an egg in serum, it won't fertilize the egg. There are two reactions. Well, there's a two-part reaction called capacitation that happens in the female reproductive tract. And the two parts that we care about are one, hyperactive motility. So basically, these tail of the sperm, which has already been activated for some time now goes into an even higher level of activation. So previously it was just wiggling. Now it's going to really try and apply some force because previously it was just trying to move the sperm. It's now trying to actively penetrate the outside layer of the egg. And so we call that outside layer the Zona Pellucida. And you can see a little bit of the structure up there in the top right. Apologies for the tiny, tiny labels. Basically, the blue and red stuff is ZP2 and ZP3. That's all you really need to know. They're this big sheet that make up in layers the Zona Pellucida, and they alternate. That's really all you need to remember about those. And they're involved in the second part. So one, we're imparting a lot more force, but force alone won't actually get us into the egg. So the acrosome reaction is what does that. The acrosome is this little, it's almost like a bit like a vesicle or something that sort of sits on top of the sperm head. And when the sperm reaches the egg, the very outside of it is going to bind to those ZP3 proteins. And that triggers a rise in intracellular calcium. Calcium is going to be a bit of a theme in fertilization, but it increases those levels, and that causes what we call the acrosome reaction. And so the acrosome basically dissolves, degenerates into lots of little hydrolytic enzymes, and those then go and they help it break down the region. And at the same time, it also that acrosome reacted sperm and now binds to ZP2. So it can only bind to ZP3 before it undergoes the reaction. It can only bind to ZP2 after it undergoes the reaction. Okay. All right, and then in terms of things that we need for this, actually, we'll talk about the rest of the of sequence next. So we've now talked about it getting through the Zona Pellucida. It's binding to the ZP2. It's making its way through. It's then going to fuse with the membrane of the actual egg itself. Most of the sperm is actually discarded because we don't need it. The sperm nucleus, the genetic information, is what's actually important, and so that enters via endocytosis, and then it triggers signaling pathways. It triggers signaling pathways within the egg, which cause the exocytosis of these things called cortical granules, which you can see sort of hanging out in the cytoplasm there. So what they do is they go through, ignore the subtitle in the diagram for Monash purposes. What they do is they cleave ZP2 and they modify ZP3, and because both of those proteins are needed for the sperm to actually get its way through the Zona Pellucida, that stops further fertilization from occurring. And there's a factor called PLC zeta, which the sperm carries, and that again, like I said, we're going to talk about calcium. That causes a rise in intracellular calcium, and that actually activates the egg. So if you look at the sort of graph of calcium activation over the course of the egg, calcium levels over the course of the egg, you first have a spike right when it tries to start making its way through. That's the acrosome reaction. And then we have a series of other spikes at sort of regular, slowly drifting intervals, which represent that activation of the egg. And then in terms of proteins, so basically there's one that each gamete needs to bring along, and if they don't bring it along, then it doesn't work. So if you have CD9 in your eggs, you're good. If there's CD9 in the sperm but not in the eggs, it doesn't mean anything. It doesn't go ahead because you need it in the eggs. You need isomo in your sperm. If you have basically you can think of it almost like a dominant gene, because if there's one functioning isomo allele in the sperm, then you're fine. If you have two non-functioning alleles, there's no protein at all, then you're out of luck, and that causes infertility basically. And so basically it's only important that sperm have isomer and that eggs have CD9. So if they have those, and then we go through our whole little process, capacitation with our hyperactivity and our acrosome reaction, and then we undergo this process to prevent further fusion, because of course that'll result in genetic abnormalities. We're pretty much ready to move on to the next stage, which is placentation. So like I said, nice and short and sweet one. Side note also, point here is that this doesn't necessarily mean it's the first sperm that makes it to the egg that actually fertilizes the egg. It's almost a bit more chance-based. So they're all going to be trying to undergo capacitation in the acrosome reaction and make their way through the Zona Pellucida. It's just the first one to actually fuse with the egg membrane. That'll be the one that actually triggers the process. But yeah, like I said, short and sweet one. And then you guys should be moving on to the next stage, which is now we have basically a viable embryo. It's going to start resuming the cell cycle, which is what we mean when we say activating the egg. We're going to make 1 cell to 2 cells to 4 cells to 8 cells, and then we're going to need to actually implant this embryo now to help it grow. Perfect. So hope you guys understood all that. Feel free to message us if you have any questions, and have a good day.